Viruses are considered to be acellular. Acellular meaning that while they do have similar structures to cells in the sense that they do have a membrane of some sort and they do have genetic material, they are considered to be non-living. They are obligate intracellular parasites. Intracellular meaning that they live inside of cells. Parasitic meaning that they require a host and they actually cannot replicate on their own. Their size, morphology, and nucleic acid composition vary depending on what type of virus they are. And we did mention a while back that while they do contain some kind of genetic material, they either have DNA or RNA. These are the only kinds of cellular-like things that exist on Earth that can have RNA independently of DNA being present as well. So they never contain both, they only ever contain one or the other. Just for a little bit of terminology, a virion is the complete infectious particle. So that would include the genetic material and any part of a viral envelope, as well as all of its specific proteins or things that it could shed, for example, to aid the virus in transmitting its genetic material into host cells. Viruses are actually very small. We had a similar picture to this earlier on in the course, but you can see over here we've got a human red blood cell, which is about 1,000 nanometers in diameter. I did mention then as well that red blood cells or erythrocytes tend to be on the smaller side. If you look at them on a blood smear slide, for example, they are non-nucleated cells, but the nucleated cells that are also present in those smears tend to be significantly larger in general. This is a bacterial cell, which we've seen at about 1,000 times under the microscope. And this is going to be smaller than a red blood cell, but much larger when we take a look at all of the viruses that are present in the world around us. But you can see that in general, they are significantly smaller than the average bacterial cell. There are many differences between bacteria and viruses, and this table is a really neat little summary of that. It shows that bacteria, for example, can be grown on artificial media like agar or in broth in the laboratory. Viruses cannot. Remember that they do require a host to live. Their division process is totally different as well. Bacteria divide by binary fission, which we've spoken about. Viruses do not. They need that host to replicate, and they have a very complex mechanism of replication compared to bacteria. Containing DNA and RNA, yes to bacteria. They do very similar things with transcription and translation of their genetic material like eukaryotic cells do, but viruses, as we mentioned, have DNA or RNA, and never both at the same time. The protein synthesis machinery, so things like ribosomes, bacteria do have, of course. Viruses do not. They use the host cell machinery instead. When they infect cells, they essentially take over those host cells. Their size we've already seen. Bacteria tend to be bigger while viruses are smaller. And sensitivity to antibiotics, yes for bacteria and no for viruses. You'll often hear doctors say when folks go into their offices demanding antibiotics for any cold that they have. If it's viral, you just have to stay home and rest and hydrate and all of that. It's because a lot of the antibiotics that we actually have target specific cellular processes, whether that's cell wall formation in bacteria, whether it's DNA replication in bacteria. Anything at all that's a normal cellular function can be targeted with medication. Viruses do none of that, so they're very difficult to target with drug treatment, though there are some ways that we can treat viruses with drugs. But antibiotics specifically are meant for biological things that are alive, right? Viruses are not. So what can viruses infect? Well, pretty much anything at all. They can infect plants, animals. There are some good examples here, like tobacco mosaic virus or the tomato spotted wilt virus. You can see that there is a physical manifestation of a disease state in these different plants. They can also infect animals. Canine distemper virus and parvovirus are two examples as well. And in addition to that, they can also infect everything else. Fungi, humans obviously, and also bacteria. We've spoken just briefly on a group of viruses called bacteriophages. They're those complex little spider-like looking ones with the head and then the legs that can inject their DNA into bacterial cells. They can readily attack bacteria. that they are meant to. There is no living thing on earth that is safe from viruses. What's interesting about them is that they are host specific. They cannot infect more than one host. We talked about this a little bit when we looked at cowpox and smallpox, where cowpox infected only cows and smallpox infected humans. So you can have multiple viruses from the same family that can infect many animals, for example, or many plants, for example, but they're going to differ in their basic structure and makeup. And as we just addressed, viruses can infect all host and cell types that exist, whether they're eukaryotic, prokaryotic, it doesn't matter which conditions of the world you're in. If you are cellular in nature, a living organic being, you can be infected with viruses. The host and cell specificity, or that compatibility I was just alluding to, is determined specifically by the interaction of viruses and their receptors. And you're going to see as we go on here that in order for viruses to be able to enter a cell, they have to have the right ticket to get in. So when they come to the surface of a cell, they need to recognize or be able to interact with proteins, certain types of lipids. There has to be some mechanism by which they can typically attach. And then after that, how the genetic material gets into the host cell will differ depending on the family or the class of virus. So when we look at viral structure, they're all very typically similar. They will have genetic material, again, DNA or RNA, and this can be sense or antisense, and we'll talk more about what that means shortly. There is typically a capsid, which is a protein coat that's going to protect the genetic material. Think of like a cell wall. In bacteria, for example, like that peptidoglycan layer. There's also the envelope, though not all viruses do have those. If a virus does have an envelope, it's normally acquired from the cell that it escapes from when they are budding. The envelope is usually made up of cell membrane components from the host cell that they infect. And if you haven't taken any biology courses yet that talk about how cell membranes need compatibility to fuse, like they have to be made up of the same types of things in order to be able to fuse with one another, this is a super important thing when we talk about delivery of materials from the Golgi apparatus to various organelles in the cell or even to the cell membrane to be secreted outward. If membranes are incompatible, they cannot fuse together. In this case, viruses will pick up the traits of the cells that they are lysing out of, so that when they meet new cells of the same type a short time later, they'll be able to fuse with them and get inside of them more easily than they would otherwise. So how do we classify viruses? Well, typically whether they are DNA or RNA viruses, that's the first distinguisher. Then we look at the morphology of the capsid. The capsids can take on a variety of different shapes. We can have those that are helical, those that are isocahedral, among other things. We look at whether or not they're enveloped and how complex they are. So a few more details on what these things mean. If they are helical, they're going to be spiral shaped or filamentous. The capsid is going to be made up of smaller units of protein called capsimeres. So you can see here that they assemble into a very large conglomerate that is coiled or spiraled in nature. So you'll see inside here the nucleic acid, whether that's DNA or RNA, depends on the viral type. And then the capsid here on the outside is made up of all of these smaller units that are coiled around into a single cylindrical form. When we look at isocahedral viruses, they are also made up of small capsimeres, but they have 20 faces. So they're much more geometric looking versus these little cylinder helical ones. But you can see that the general intent is the same. The capsid, made up of these small units, protects the genetic material that is internal to it. And in terms of testing, when I ask you about these kinds of things, I'm not going to be asking you, for example, what kind of virus is the tobacco mosaic virus. So don't worry about the examples here, but try to focus more on what types of physical characteristics, what kinds of traits distinguish these different groups from one another instead. We also have enveloped viruses that do have that envelope that we discussed, which again is primarily lipids and proteins. And internally, you can see that capsimere layer with the genetic material internalized and protected. And you can see down here bacteriophages, which we already mentioned, that have this very distinct and unique anatomy. That's why they're called complex. They're not just a ball of protein with some genetic material inside, but they have an entire interesting and developed mechanism that's different from a lot of viruses in terms of how they get their genetic material into cells. The tables that are going to follow on the next couple of slides are just for your information only. I have no expectation that you'll spend time memorizing all of these different families. But one thing I just wanted to point out here was where coronaviruses fall. So they are single-stranded enveloped helical viruses. Obviously, they're in the forefront of many of our minds these days. So there they are. Another thing to mention here is that RNA in viruses can be double-stranded. This is something that does not happen normally in other organisms. RNA is always single-stranded because it's made from one template of DNA. And if there is double-stranded RNA present in eukaryotic cells or eukaryotic organisms that have an immune system, those organisms can recognize double-stranded RNA as foreign and can target and eliminate it because it is such an anomaly when we look at the general occurrences of RNA in most organisms. We also have a large number of DNA viruses as well. So just take a look at these. Maybe some of these names feel familiar to you. Again, just for your information, of course. In your notes, you'll find a couple of slides that will follow that break down some of these group members into some more detail. And again, that's just for you to have to read over if viruses are something that interests you in particular, but I won't be testing you on any of that material. So here we have another great diagram of an enveloped virus. You can see that at the most internal portion, we have DNA or RNA, then surrounded by a capsid, which is again that protein layer, usually made up of capsomeres. And then there is the viral envelope, which is made up of proteins from the host, like I've already mentioned. The surface proteins are something that we're going to start talking about in a bit more detail here. And these are what allow for viruses to interact with cells that they're compatible with in order for them to be able to infect a specific type of host cell. You'll notice that viruses, even within a complex eukaryote, will always only infect the same cell type. It's not as if you get a virus and it goes and infects every single cell in your body, because your different cells are going to express different things on their cell surfaces. So the surface proteins that are present on these viruses only allow them to interact with cells that they can target. There's many steps in the viral replication cycle, and we're going to talk about each of these over the next couple of slides. So depending on the viral type, the mechanism will differ slightly, but in general, lots of these things do happen in most viral replication cycles. The attachment portion is obviously what we were just kind of mentioning. So the receptors on the host cell have to be compatible with the proteins that are present in that viral envelope. So the host cell has to be able to recognize or bind to the virus in order for them to be able to get their genetic material into the cell. There's no drive-by viral interactions with hosts. They have to be able to attach. And this, again, allows for that specificity between viruses and their host cell targets. When we look at the difference between fusion or penetration, these are slightly different. The membrane fusion can be where you have the cell envelope of the host fusing with the envelope of the virus, which would essentially allow for the contents of the virus to pass inside the cell very easily. Or there's endocytosis. Endocytosis is a type of phagocytosis where the cell membrane would detect a virus after it's attached, extend its cell membrane around it, and take it in. So two mechanisms. Once inside, nucleocapsids get uncoated, or alternatively, the genome can be directly injected inside of the cell. And we did talk on that lightly with bacteriophages. They have a little pin that penetrates the cell wall and membrane, makes a little channel, and directly passes that genetic material into the cells that they are infecting. So this here shows fusion. So you can see that there's an attachment step, as we've already discussed. And then as that envelope starts to meet up with that host cell membrane, because they are made up of identical things, right? Because this envelope came from another virus that lysed out of another host cell that's of the same type and kind. So you do have that fusion event where the envelope will become one with the membrane of the cell that it's infecting. You can see here that fusion is starting to unify these two entities, where the internal portion of this virus is becoming open to the cytoplasm of the host cell. And eventually you'll see that all of those envelope lipids and proteins become part of the host cell membrane. And then this is that nucleocapsid that has been released. And that genetic material from that virus is now inside of the cell. If this was endocytosis, you would instead have this attachment step, and then the elongation of the membrane, it would come up and around this viral particle as a whole and engulf it. So slightly different, but still requiring that attachment. For viruses that are non-enveloped, they're going to come across the plasma membrane directly. And typically that'll be done through endocytosis as well. So you again have that extension of the cell membrane around this particle, because don't forget, cells are always kind of monitoring their environment. If there's something that comes and attaches to them, typically they engulf it to see what it is and to assess it, which is a good thing normally, but a bad thing when it comes to viruses, because once the viral DNA or RNA is inside, then they can set their self-replication cycle into motion. Once inside the cell, the virus has to uncoat. So the uncoating step dissolves the capsid or disassembles it to reveal the genetic material inside. To make more copies of itself, it has to obviously expose its genetic material, because this is where all of the information for making new viruses is going to be kept. Viruses that are uncoated inside of a host are said to be in the eclipse phase, which lasts until all of our newly synthesized viral particles emerge from the host cell during the lytic portion of the viral replication cycle. In terms of the synthesis step for the genome, most of it is carried out by host cell machinery. So in the case of DNA viruses, that's going to be taken care of by host cell machinery, because our DNA is also double-stranded. So that's an easy stretch to make for viruses. For RNA viruses, though, they have to have their own enzymes encoded in order to replicate their own RNA, because we don't typically do that. They'll also need to synthesize new capsid proteins for new viruses, and also replication enzymes for future generations of them after they get released and go on to infect their own host cells. So no matter what type of genetic material a virus has, essentially to get themselves to mRNA in order to get new proteins made for future generations of viruses, they have to undergo several different steps. So no matter what a virus is, they do have a means to make their genetic material work in order to replicate themselves. So these examples here kind of encompass all of the different possibilities that viruses can have, where they start with single-stranded or double-stranded DNA or RNA. The protein synthesis of the new viruses is always going to occur in the cytoplasm of the host cell, which is very typical for protein synthesis in general. For assembly and release, all of the parts of the virus that have been formed, so the replicated genetic material, the translated proteins of whatever is required for that specific type, assemble together to make a complete virus. Depending on the type of virus, this can happen in conjunction with a membrane, or it can happen in the cytoplasm as well. The release of viruses can either be by budding or by lysis. If it's by budding, it's kind of like the reverse of endocytosis or phagocytosis, where the cell membrane will just kind of pinch off a little bubble with a virus in it, and then that virus can go on to find a new host cell. Or the alternative is lysis, where so many viruses replicate inside of a cell that the cell will burst. Those bursting events release lots of synthesized and assembled viruses into the immediate environment, where those new viruses can then go on to find new host cells to infect and repeat the cycle. So to break down sense and antisense a little bit, single-stranded RNA can be either or, either sense or antisense. If something has sense RNA, it means that it's already in a configuration where it can be directly translated. So nothing has to be done to it in order to make it translatable to become protein. There are a number of RNA viruses that have antisense RNA, which is where they have a template sequence that has to first be transcribed into a sense strand. And because we don't typically have enzymes in our cells that can make an RNA template, we can do DNA just fine, but RNA not so much. They have to carry their own RNA-dependent RNA polymerase, which is a protein that will synthesize RNA from existing RNA. For those viruses that are double-stranded and have RNA as their primary genetic material, they're going to carry both sense and antisense strands, as you imagine, right? You have your template strand, and then you have your sense strand, and both of these are going to play their own roles in viral replication in those cases. I think the basic information on the previous slide was plenty for our level of exploration of viruses, but this diagram just serves to break down the different types of sense or antisense RNA mechanisms in RNA viruses. So while I'll leave this in, you're welcome to kind of sit through if you need a little bit more of a further explanation on some of these things, but this is not something that I will be specifically testing you on. You just really need to know the difference between sense or antisense and how that will make the replication cycle different. We're going to talk about some different viral examples now. And the first will be retroviruses. You've probably heard of those, or antiretroviral medications. And a good example of this is HIV, or human immunodeficiency virus. These are viruses that have single-stranded RNA that have to essentially go backwards from the central dogma of how most genetic material runs. Think about every other cell that you've ever heard about. You have DNA that gets transcribed into RNA, and RNA is translated into protein. For retroviruses, they actually have a mechanism that requires them to go backwards from RNA to DNA. So they have a group of enzymes that allow for that to happen that is abnormal in every other living thing that has ever existed. The protein that they carry that allows them to do this is called reverse transcriptase, which is specific only to these kinds of viruses. So you can see in this example, you have the HIV virion that is going to come and attach to the cell that it's infecting. If you've forgotten, these affect immune cells called CD4 or T helper cells. They bind to a specific receptor on the cell surface called CCR5. After they've bound, they have their RNA, which gets released into the cytoplasm. And then there's reverse transcription into DNA. Following that, there's that integration step that hijacks all of the cellular machinery to produce more proteins to form new HIV particles that eventually do get released through budding, and the process continues over time. Because reverse transcriptase is such a unique and special thing, this is how we're able to slow the progression of HIV in patients with this specific type of virus. Because if we're treating and blocking reverse transcription, we're not affecting any mechanism that's normally found inside of the host. This is only specific to this viral type. DNA viral replication is a lot more simple because obviously our cells have DNA in them and that's their normal mechanism of function. So if a DNA virus fuses into a cell, you end up with intact genes that our cell knows how to deal with. We can create mRNA from the DNA sequence, which can then be translated into protein and eventually form those new virions to be released. Much simpler overall, just because the host cell machinery is well adapted to this type of thing. When we look at bacteriophages, we're looking at bacterial cells as hosts. Bacteriophages do not infect eukaryotes, but you can see here that we have the bacterial cell wall, the chromosome inside. We've got the bacteriophage that is going to land on the surface of the bacterial cells. And you can see here that these little legs of theirs give them some flexion on the cell's surface. And after that, they're going to penetrate the host. So we did talk about that, where they release a little tube that comes through the cell wall and the cell membrane to allow for the release of their genetic material into the bacterial cell. Following that, there's that typical replication step, hijacking all of the stuff inside of the bacterial cells to allow for those replications to occur. And then there's the synthesis and release of the bacteriophages into the environment. So you can see here, this is the maturation and assembly step, where all of their little parts are getting combined to make a new bacteriophage. They're quite complex. They're very interesting. They're very, very distinct under the microscope as well. And then typically bacterial cells that are infected with bacteriophages will lyse and release the new bacteriophage particles into the environment to go and infect new bacteria. Now, we often talk about the lytic cycle of viral replication, but there's also the lysogenic cycle. And the difference primarily is that the lysogenic cycle is almost like a dormancy state. So if we take a look at this here, where we do have the infection of a cell with a virus, sometimes that DNA is used right away to replicate and create new viruses and release them to perpetuate this cycle. Occasionally, if DNA or genetic material is injected into a cell, what will happen is that that sequence will get embedded in the host's genome and will be kept for many, many generations of cells. Many, many cell divisions later, that virus may very well want to pop back into existence. And there's a triggering event of some kind that will cause the transcription and translation of these embedded portions of DNA to form new individuals many, many divisions later. Another thing that can occur from the lysogenic cycle is a conference of virulence to bacterial cells that have incorporated these genes into their genome. Bacteria that have viral genes can, in some ways, have more potency or disease-causing abilities based on this new genetic material that they have inherited. This is observed in a couple of different situations, like cholera toxin, for example, that's produced following the infection with the CTX phage in Vibrio cholerae. And similarly, when we think of diphtheria, the toxin that's produced in that illness is also phage-induced. So there's almost like this symbiosis that can happen sometimes between viruses and bacteria that can result in even more powerful bacteria that can cause broader and more impactful diseases in human populations or other populations. So why necessarily would we want to cultivate and study viruses? Well, because they affect humans especially, but also so many other organisms, it's important to know how they function. We want to learn about their replication strategies. We need to uncover how they interact with host cells in whoever they happen to be infecting. We have to try to determine to the best of our abilities what will kill them if they do cause disease, and develop new antiviral drugs or potentially vaccines against different types of viruses that are causing a lot of problems. So as we know, viruses cannot be grown independently in the laboratory. They have to have some kind of living host. So there are a couple of mechanisms to do this. You can use embryonated chicken eggs, where you can inject viruses into something undergoing gestation to raise up viruses and to study them. Or you can use cell culture, where you're growing cells independently of a host. This is called in vitro work, where you have cells from living organisms, but that grow independently of them. So it's done synthetically, but the cells are still living. They don't necessarily behave the same way that they would if they were still part of their original organism, but they still do have most of the traits, at least, that they would have in terms of their protein makeup, their genes, their type in cell culture. When we want to detect and measure viruses, we can do something called a plaque assay, where viruses get plated on cell monolayers. So you have cells that you are growing in culture, and you grow them until they are forming a single layer on the surface, in this case of a small well. So this is a plate with many of these little wells or depressions in it, and you plate cells in these wells, and then you infect them with viruses. When the viruses infect these cells, if they do the lytic cycle, they'll cause them to lyse. And you'll see zones of clearing, which will equal plaques. And the plaques are shown here as these little white dots that appear in the otherwise homogeneous blue. The viral titer, or how many viruses are present in a person, can be calculated based on the number of plaques that are found in a specific assay. So it's a really good way to tell if somebody is infected, for example. If you're trying to see, does somebody have this virus, and the virus does go through that lytic cycle, you can do this as a great test to assess whether or not a virus is present. Or if you're trying to calculate how virulent and how destructive a virus is, you could set a time point on this. So how many plaques occur over a specific time frame? How many plaques occur over 6 hours, 12 hours, and compare different viruses to one another to get an idea of how fast a virus can work. You can also use another process called ELISA, and this is an enzyme assay that uses antibodies. Antibodies are proteins that are very specific for their antigens. And viruses obviously have their own set of proteins, whether they're on their envelopes, whether they're things that are getting produced inside of the cell during the replication step. Either way, they are going to be considered specific only to those viruses, and we can use antibodies that are conjugated to some kind of indicator. The indicators can produce color, they can produce light. So just to simplify this, if there are viral antigens present and you add antibodies to them and they bind, that means that they're there. So you can, again, diagnose if a person doesn't have a virus and you add these antibodies with these indicators in them, they won't bind to anything and you're not going to see the light or the color get produced. So it's a good way to give us a yes or no, is it present or not, in clinical samples, for example. There are also some viruses that have proteins in their envelopes that can cause other cells to bind together, specifically erythrocytes. If those viruses do contain those proteins, and you add them into an assay with red blood cells, what they do is they cause a lot of red blood cells to clump together, similarly to what we see when we do blood typing. So what you'll see is hemagglutination, where you've got deposits of little clots of blood cells that form in samples where viruses are present. In samples where there are no viruses, you're not going to see that clumping. So this is again a great yes or no assay. Does somebody have this particular virus or not? Viruses are also very visible through microscopy. With scanning electron microscopy especially, if you are to visualize parts of the cell, you can definitely see the shapes of viruses. And what's interesting about them is that they're all visually distinct. They all have their own different types of shapes, sizes, configurations that allow us to be able to look at a slide of them and say, oh, that must be a pox virus, or oh, that must be Ebola. They're all very different and distinct from one another. In this example here, you can definitely see that the cell looks different. When we've seen DAPI dye in other examples earlier in this semester, the nucleus is uniformly blue, but in this case, you can see that there are many viral particles present. So a great snapshot of a virally infected cell can again be diagnostic, or it could be used in a variety of other ways to assess some property of viruses. And here's a nice photo of that electron microscopy I was just alluding to. You can definitely see that the viral particles are visible. They are able to be identified positively. And again, they will look very different depending on what family of virus has infected the host that you're looking into. Now, occasionally there will be viruses of interest. So, for example, at the start of any outbreak of a virus, people will want to know what it is, right? If people are getting sick and a lot of people are exhibiting signs of disease, there may be an interest in getting to know what a virus is, and that will be done through sampling the source of interest. What you can do from there is isolate that genetic material. You can isolate the proteins. You can start essentially breaking down that pathogen bit by bit in order to identify what type of virus that it is. And you can see in this example here, there's the isolation of RNA or DNA from one of many different sources of interest. And then you can try to amplify that DNA with PCR to be able to compare it to known libraries of viral genetic material, or you can do mass sequencing as well. But through either mechanism, you're assessing the actual sequence of that genetic material to try to classify it. We'll cover now a couple of concepts in viral infections here. Pretty much your viral entry sites can be through any tissue that's compatible with the virus. So typically, most infections get into hosts based on their common portals of entry. So these are areas of the body that are exposed to the environment. They include the skin, respiratory tract, the genitourinary tract, gastrointestinal tract, or you can have events that will penetrate the skin barrier and inject things directly into the bloodstream. Infections can be of two general types. You can have acute ones where there is a rapid onset of the disease. There's a relatively brief period of symptoms that resolve quickly, and there are no long-lasting effects of an acute infection. On the other hand, you can have chronic infections. These are not self-limiting, meaning that they don't end. They perpetually stay in a host. Symptoms are long-lived. They fail to resolve. They become persistent. And a good example of this is HIV, where, as we discussed before, there isn't a true and tried mechanism that exists either through chemotherapeutic intervention or immunity and our own immune system's function that can truly rid the body of this infectious agent. You can see differences in how infections spread. There's a primary infection where the infection stays localized at the site at which it was introduced. Or you can have systemic infections where things will hop out of that area where they were primarily introduced and spread either by the lymphatic system or the blood system. A good example of a systemic infection is pox diseases. We're probably more familiar with chickenpox, where you get widespread lesions that form all over the body after you become infected. The pathogenesis of a virus is going to differ depending on the type. Pathogenesis simply means how much disease is going to be caused via virus. It'll depend on the mode of entry, how much cell damage occurs, how much lysis is occurring in those hosts, how much of that tissue that is targeted is being damaged. Is it being repaired faster than it's being damaged, or is it being damaged faster than it's being repaired? The host responds. Are we able to control it? We have an entire leg of our immune system that actually goes and seeks out to destroy infected cells. This is facilitated primarily through a group of cells called cytotoxic T cells. Cytotoxic meaning cell killing. They can detect cells that are abnormal, and abnormal cells that have been infected fall under that category. And then finally, the clearance or the persistence. How difficult is it to get rid of this virus from the host? Can it be gotten rid of entirely? All of these things will factor into how disease-causing a virus is and how detrimental it is to the host that it affects. Protect yourself from influenza is the thing that we would normally be saying around this time of year during flu season. And obviously at this place in time, another thing that you could stick on this sign would be protect yourself from coronavirus. We're going to talk a little bit here about influenza, a couple of examples of viruses that very much impact our day-to-day. Influenza is going to be an enveloped virus from the orthomyxovirus family. It has an RNA genome. The two major types of glycoproteins that it has in its envelope are hemagglutinin and neuraminidase. You may have heard, oftentimes, viruses being called as an H number, N number. H1N1, H1N5. That refers to the hemagglutinin and the neuraminidase proteins that are found in the cell envelope. And if you take a look at the viral anatomy here, you can see that they've shown here neuraminidase and also hemagglutinin. So those are just envelope proteins that are particularly associated with influenza. There are 16 different types of hemagglutinin and 9 types of neuraminidase that can be produced natively by these viruses. And different combinations of these proteins result in different variants, right? So they're all influenza, but depending on what they're expressing on their cell surfaces, they're slightly different from one another, if that makes sense. When we look at the transmission of influenza, it's always going to be by aerosols. So people coughing, speaking, talking, sneezing, it doesn't matter. All of those are going to cause droplets to form in the air. We're seeing a lot of this same discussion around coronavirus with masking. Masking prevents the travel of these aerosols for long distances. So masking, while it doesn't necessarily completely prevent viruses from getting into the environment after a time, it certainly limits how far they can go. And if everybody is masked, obviously, you're going to not have as much of that transmission from person to person as you would if people are not wearing masks at all. New strains of influenza come around year over year. And because of this, we are forced to vaccinate year over year as well to keep up with new variants of the disease that are being produced. Lots of what we see is caused by something called antigenic drift, where minor changes in viral structure are occurring. You could have a slight mutation on one of those hemagglutinin or neuraminidase proteins. You can have a differential expression of those, but every once in a while, we end up with what's called antigenic shift, where there is a major reassortment of viral genomes. That could be by fusing with another virus. It could be some kind of mutation that causes the expression of something that is a lot more virulent than the original form, but this causes strains of that virus to be more disease-causing than other variants are. This is why every couple of years, for example, we see an influenza outbreak that is more problematic than it has been in previous years. And this is due to antigenic shift.
Pandemics, or global epidemics of influenza, have occurred for hundreds of years. There were 3 pandemics during the 20th century. They occurred in 1918, 1957, and 1968. The 1918 pandemic took the largest toll, causing an estimated 50 million deaths worldwide. The first pandemic of the 21st century began in 2009. But how does a pandemic occur? Influenza A viruses vary slightly from year to year, a phenomenon called antigenic drift. These small variations are the reason why the vaccine must be updated every flu season. Influenza A viruses also can undergo dramatic genetic changes called antigenic shift that may cause a new influenza strain to which we have little to no immunity. If this strain spreads easily among people, it may cause a pandemic. The immune system targets 2 proteins on the surface of influenza A viruses, hemagglutinin and neuraminidase. Hemagglutinin has 16 confirmed subtypes of proteins. Neuraminidase has 9. From these protein combinations, scientists name influenza viruses. For example, an H5N1 virus contains hemagglutinin subtype 5 and neuraminidase subtype 1. Antigenic shift can occur when two different influenza A viruses circulating in the environment infect and multiply inside the same cell. While inside the cell, these parent viruses can exchange or reassort genetic material and create a new virus. Through reassortment of the 8 gene segments of each virus, there are theoretically 256 possible virus combinations. Pandemic viruses also can emerge in other ways. Among these is adaptation. Scientists believe that the 2009 H1N1 virus was the product of 2 influenza A viruses that were circulating in pigs. The viruses are thought to have reassorted and adapted to create a new virus capable of infecting people. If an emerging virus transmits efficiently from person to person, it would be a challenge for scientists to produce an effective vaccine fast enough to limit its spread. That is particularly true with the amount of international travel that occurs today. In a matter of hours, one person infected with a new form of influenza can spread the virus to people in another part of the world. The National Institute of Allergy and Infectious Diseases and other research institutions around the globe are working to find better ways to prevent, diagnose, and treat pandemic influenza. These efforts include testing new drug and vaccine concepts, studying flu viruses in animals and humans, and documenting factors that could lead to the emergence of strains with pandemic potential. The aims of these projects are to help health officials quickly detect an emerging influenza A virus and implement measures that bring the outbreak under control.
We'll shift gears here and talk about Ebola now, which is another virus that has had several epidemic outbreaks in various areas of the world over and over again for many years in the last century. This is an enveloped filovirus, single-stranded RNA genome. It has a filamentous structure, so very different from what we've seen in a couple of other examples. And transmission occurs in these cases through close contact with blood or other types of bodily fluids. Bats are thought to be the natural hosts of the virus, and bats tend to act as a reservoir. So you'll see Ebola disappear for many years, and then human populations will come into contact with bats again, and Ebola will reemerge in human populations. So it tends to not hide away, but it tends to not affect humans for many years in between exposures. In terms of Ebola's mechanism, the virions are going to fuse with cell membranes, after which the virus uncoats, and the acidic lysosome, which typically kills things that get engulfed by cells, is not enough to destroy Ebola. Ebola persists in the lysosomal environment and actually replicates there. So this is a diagram that shows some Ebola virus ecology and how it actually sits in different populations within an ecosystem and can eventually come back and infect humans every so often. So you can see over here, we've got different bats that are replicating, they're passing Ebola around, but bats are not affected by it. They just carry it, so they don't normally die from it. Sometimes bats will come into other populations of animals that may come into regular contact with humans, either as a food source or just be around in the general day-to-day. And then once that human contact occurs, the virus is transmitted into humans and then transmitted further between humans, and it can spread very quickly through those populations and impact them very negatively. Fatality rates for Ebola range from 50 to 90% because it mainly affects human immune cells and endothelial cells. If you're wondering what endothelial cells are, they are the cells that line your blood vessels. So if Ebola is infecting those cells and lysing them, the tissue damage in your blood vessels can cause leakage and, as a result, hypotension, because your blood pressure will drop as your closed cardiovascular system is no longer considered to be closed, which can in turn result in death because you're not getting enough blood around the body with that decreased blood pressure. Viruses have also been linked to several different kinds of cancer, and these are called oncogenic viruses. Oncoviruses or tumor viruses can lead to cancer in a couple of different ways. They can activate what's called an oncogene, Oncogenes are genes that are linked to different kinds of cancers and are typically expressed only in situations where a cell has become cancerous. And this can be done by DNA integration. Our cells have a number of genes that are expressed regularly to help suppress irregular cell activity, because at its core, cancer is when cells kind of go off the rails in terms of their normal cell division processes. Cells are not allowed to just divide uncontrollably and do whatever they like. We have genes that are present to create proteins to put the brakes on and to allow for cell division to be very highly regulated. If a virus happens to insert its gene in the middle of one of our genes that codes for some of those proteins that put the brakes on, that's when we see a loss of what are called tumor suppressor genes, where all of a sudden the mechanisms by which we control cell division and normal cellular function are lost. And that can cause cells to not have those regulatory mechanisms and become cancerous as a result of that. These viruses can also have proteins that activate oncogenes. So, a set of genes, for example, that are only turned on in cancer states, so they can flip that switch in our cells and push them into those mechanisms to drive cells to become cancerous when they're normally not. Some great examples of these are human immunodeficiency virus, Epstein-Barr virus, human papillomavirus, and there are many, many others. We'll take a look here now at human papillomavirus, which is a virus that's been heavily implicated in the development of cervical cancer. And what it does is, following infection of a host, the HPV virus can infect cells of the cervix. And so you can see these basal cells are impacted. Over the course of weeks, you're going to have replication of that virus to create new viral particles. Some people heal up and they're absolutely fine following infection. But in 10 to 30 years, as this goes on and on and on, what you end up with is that integration, which can disrupt some really important genes for tumor suppression, which can cause irregular cells to become predominant and eventually cause cancer in that tissue type. A very brief little bit here about prions. Prions are not considered to be viruses, but they're another type of disease-causing acellular entity. They were originally thought to be viruses because there is a fairly long incubation period for them following infection prior to seeing symptoms emerge in infected individuals. But viruses at least have some kind of genetic material. Prions are just simply proteins. They're small peptides, actually, like small lengths of proteins that can particularly affect the neurological systems of their hosts. They are very transmissible, and we've seen examples of this a lot of the times in livestock. So things like mad cow disease, Creutzfeldt-Jakob disease, and scrapie. And you can see the manifestation of scrapie here in this poor ewe that has lost a lot of wool from scratching due to its infection. What prions do at their core is cause the misfolding of proteins. And when you have dysfunctional proteins, you're going to have a loss of function to certain extents in the host that it's affected. This can lead to neurodegenerative disorders if your cells are not working properly or they don't have the correct machinery in order to do their jobs well. And while there's still lots of great research being done on prions, they're probably one of the least understood disease-causing agents that currently exist in our world today. So I think we'll leave it there for now, and thank you all very much, as always, for your attention. If you do have any questions, please feel free to contact me at laura.r@nipissingU.ca, and I'll see you in the next one.